Hello and welcome to Dr. Varsha Gaitonde's channel. In this video, we shall continue the discussion on current affairs, MCQs in GPB and some of the other general topics. Our first topic is USDA approves GM purple tomato for cultivation. As you generally know, tomatoes used to be orange or generally red in color, but here the purple color denotes the richness of anthocyanin content in the tomatoes. After over 14 years of development of the purple tomato, the GM crop has been recognized as safe to grow by US Department of Agriculture. The seed of the plant developed by Norfolk Plant Sciences that may enter the market for sale by the year 2023. So here two things are important: for what purpose this purple tomatoes are developed and the gene involved in it. As I already told you, this is for high anthocyanin content. This content of purple fruit from a hemizygous Del Bar Rose 1 plant averaged approximately 500 mg per 100 gram fresh weight, whereas anthocyanin were undetected in wild type of red tomatoes. Our second topic is about Samba Mysuri, improved Samba Mysuri. Indian scientists gene edit a popular Samba Mysuri rice to achieve an impressive 35% yield increase. As you know, Samba Mysuri is very well known for its grain quality, fine grain quality, which is very popular in central as well as southern India, where the rice consumer population is high. As Samba Mysuri is having some of the lacunas like susceptibility to bacterial leaf blight and other characters, this the popularity decreased gradually, but after that by marker assisted selection and all, they have improved its lacunas, means they have overcame its lacunas. So improved Samba Mysuri was by marker assisted selection technique, and now again gene edit technique are imposed on it. So Samba Mysuri is nothing but our BPT 5204. Here this plant, generally Samba Mysuri plant gives 200 to 250 grains per panicle, whereas genome edited plants give over 450 grains per panicle. Our third topic for discussion is understanding GM mustard. As you already know, the Barnes and Barster gene system in mustard. This is a very old topic. The efforts of Delhi University scientists Deepak Pental and Bist, where they developed the Barnes Barster system, incorporated bar gene by genetic engineering approach. Here, for your better understanding, I'll just draw the Barnes Barster system to recall your memory. What will happen? First, the bar gene. You should know the source of bar gene, that is from Bacillus amyloliquefaciens, source from where it has been taken. That is a soil bacterium. Okay. From there, what they did, they took the transgenic plant with Barnes bar gene, then expression of Barnes bar gene in plants. After expressing, what they will do, this majorly work on tapetum, that means it will inactivate the male reproductive system Barnes, that causes the male sterility. So what we will obtain, we will obtain the male sterile plants. Okay, so after that, when we cross it with the normal or untransformed plant, will get the 1:1 male sterile and male fertile plants. Now the second system comes, that is Barster system. What is Barster? That will restore the fertility of Barnes bar system. Okay, so to obtain the F1 or the hybrid seed which are fertile and which will give fertile seeds in next season, we should cross at any cost with the barster system. So here whenever we are obtaining 1:1 system, that is sterile and fertile, if we will spray the herbicide here is, you should tell me in the article also I have mentioned here. When we spray it, what will happen? This fertile plants will die, so that only sterile plants will be obtained, and for that we'll cross with the barster system and we get the F1, which are fertile with Barnes bar gene. Okay, that is all about Barnes barster system in GM mustard. So why now it is in use? The hybrid mustard, Dhara mustard hybrid 11, is in use for its better yield. Hybridization is very difficult in mustard because as this particular species is self-pollinated, where hermaphrodite flower, that means male and female are at the same position. Hybridization or the emasculation, bagging, tagging, whatever is there is very difficult, and by incorporating or by imposing this GM system, there is a drastic improvement in the hybridization program, so that we can increase the oil content or we can increase the yield up to 28%. So one more article is there, GM mustard will be ready for cultivation in 3 crop seasons. As I have already told you that this is not a new approach. This took almost 14 to 15 years or more than that for research, but to accept and adopt in our system it took a long time to get approval or to get acceptance by GEAC, that is Genetic Engineering Approval Committee. It took a long time. Still there are so many oppositions as well as acceptances. So why government is giving so much emphasis on GM mustard? Because as you know, due to Russia-Ukraine war, the oil import has drastically reduced, and we are majorly dependent on oil imports for our growing population, to feed our growing population. So somewhere this GM mustard will certainly help us to maintain the oil prices and all. So, our next topic is Apis karinjodian, a new species of bee discovered in India. This is a general topic, but still very important. As I was discussing about GM mustard, many of the environmentalists told that mustard is somewhere having very attractive flowers, and the GM or genetic engineering may affect the pollination or honeybees reproduction and honey production, and somewhere the consumption of honey may affect us. But remember, honey is purely a sucrose or sugar content, and the Barnes bar system is completely a protein based approach, so there won't be much effect on honey production or honey consumption. So coming back to Apis karinjodian, this was discovered in Kerala with the efforts of a team consisting uh, Shanas S. Assistant Professor of Kerala Agriculture University, then Anju Krishnan and other co-workers, we may say. This is called as Indian black honeybee. So this new species is endemic honeybee, has been discovered in Western Ghats. This is named as Apis karinjodian and given the common name black Indian black honeybee. Okay. Our next topic is paddy residue utilization. This is recently in so much news because of the increasing the rate of Delhi air pollution and NCR region pollution. There are two approaches to reduce and to utilize the rice paddy straw in a better way. Those are torrefaction and pelletization. So first we should know what is pelletization and torrefaction. Pelletization is a process of molding biomass into dense pellets, whereas Torrefaction is the process in which biomass is heated in oxygen deficient environment to completely remove the moisture. By that, what they'll do, they'll feed the cattle, and it is utilized for different purposes like mushroom cultivation and for preparing some hay stocks and all those things. Why it is in news? Because the Union Ministry of Environment has announced one time only scheme to incentivize the industrialists to set up paddy straw pelletization and torrefication in plants. The union government will fully fund these two processes with rupees 70 lakhs subject to capacity. The maximum funding will be 1.4 crores. Next topic is edible oil from algae. New source of cooking vegetable oil has been discovered from algae. Scientists found way to extract oils from algae which can be used in the same way as vegetable oils. This is a significant discovery as algae are one of the most plentiful organisms on earth, can be grown in a variety of environments. New oils are also more environmental friendly than other vegetable oils as they do not require large amounts of lands or water to produce. In addition, process of extraction of oil from algae is much less energy intensive than other methods. The new oils are expected to be available to consumers within next few years. The approach is done with the help of microalgae, which is considered an interesting candidate for the sustainable production of edible oils because microalgae contain abundant polyunsaturated fatty acids, that is PUFA, which is good for our heart and have high photosynthetic rates and high oil productivity. They are superior to terrestrial oleaginous crops for producing edible oil. That means less space. With the industrial approach and applications we can produce it, and within few years we may get these algal based edible oils in our market. Next topic is most popular GI tags. In plant breeding perspective, GI tags are very important. We should always update the recent GI tags into our notes. So Hyderabadi Halim has won the National Intellectual Property Award of 21-22 under the most popular geographical indication of India category under food stuff category. This was in a competition of about 17 food items with GI status from across India, including Rasgulla, Bikneri, Bhujia, and Ratlami Sev. In 2010 only, this Hyderabadi Halim got GI status, and in 2022, the Registrar of GI renewed the tag for the Hyderabadi Halim dish for next 10 years. The award for most popular GI of India are presented under five categories, that is agriculture, foodstuff, handicrafts, manufactured, and natural. Now we shall see some other category most popular GIs awarded this year. Under agriculture category, Kandamal Haldi from Odisha, handicraft, Tanjavur art plate from Tamil Nadu. Under manufactured GI section, Mysore sandal soap from Karnataka, and under natural GI category, Chunar Bhaluwa Patthar from Uttar Pradesh. Now, mock wheat. Mock meat means this is a kind of vegetable based meat. Recently, demand has been surged in India. The term used for alternative meat options because being vegan became a popular theme and caption these days. It is composed of plant based proteins processed to have a meat like feel, texture, and taste. This particular production takes or uses wheat gluten, soya protein, or vegetable substitutes like jackfruit. Remember these things: wheat gluten, soya protein, or vegetable substitutes like jackfruit are utilized here. It is different from lab grown meat, which is cultured meat cultivated using animal cells collected from animals, available in ready to eat and ready to cook forms. Next is Nihonshu. As we were discussing about GI tags, Japanese assembly has applied this nihanshu, an alcoholic beverage made from fermented rice. Next is Pokkali rice. This again became a general topic. This is a saltwater tolerant paddy endemic to central Kerala. Why in news now? Because due to fish based uh, disruption in crop calendar, this particular rice is the cultivation is affected drastically. This GI tagged rice is endemic to central Kerala, that is grown in the coastal fields of Alapazha, Ernakulam, and Thrissur districts. It is raised between June and November, followed by a season of fish farming. After paddy's harvest, the stubble in the fields act as food and shelter for shrimps and small fishes. Fish excreta and scales, along with the decomposed stubbles, provide excellent natural manure for another season of Pokali. However, farming tradition is under threat due to the fish farmers and shrimp cultivars disrupting the crop calendar, yield, high labor cost, and poor returns. Now, government releases guidelines to speed up development of plant varieties. Why these guidelines came? Because nowadays the science field or the agriculture field is moving fast with the speed breeding, genome editing, genetic engineering, marker assisted selection, association breeding, and all those approaches. The guideline shall be applicable for all organizations involving research, development, and handling of the genome edited plants under site directed nucleases category, that is site directed nucleus 1 and 2. They are expected to speed up development of plant varieties and reduce the time for approval. That means, as you know, whenever to approve the GM food crops, when it comes to GM cotton or GM brinjal, it may be, there were so many efforts to approve the GM brinjal, but it could not happen. So it takes so much time to approve those. But genome editing is somewhat different from genetic engineering. This SDN technology helps to introduce specific small changes at the site of DNA breaks. So this is not genetic engineering. The genome edited plants derived from SDNs are generally classified under three categories, that is SDN 1, 2, and 3. Under 1, here it is used without using DNA sequence template, whereas in SDN 2, a site directed mutagenesis using a DNA sequence template, and in SDN 3, site directed insertion of gene DNA sequence using a DNA sequence template. SDN 1 and 2 do not lead to the insertion of foreign DNA. So these were the general topics as well as some of the topics related to genetics and plant breeding. Now we shall discuss some of the multiple choice questions. Our first question of the day are: Mendel studied trait inheritance, gene inheritance, variation inheritance, and genetic inheritance. The answer is trait inheritance. As you know, Mendel never described a trait, but he told about the factors. So factors are nothing but the traits. So he talked about the trait inheritance. Polyploid plants can arise spontaneously in nature by: the options are meiotic failure, mitotic failure, fusion of unreduced gametes, and all the above. Answer here is all the above, because polyploids are generated by meiotic failure, mitotic failure, fusion of unreduced gametes, and all these approaches. Next question: consider about specification via polyploidy. The first one is: a diploid cell undergoes failed meiosis, producing diploid gametes, producing triploid gametes. Fourth option is: which self-fertilize to produce tetraploid zygotes. Means speciation via polyploidy. Speciation means species generation due to polyploidization approach. How it happens? They have given different approaches here, and the question is to choose which are the correct one. So here 1 and 2 are true, 2 and 3 are true, 1, 2, 4 are true, or only 1, 3, 4 are true. The answer here is 1, 2 and 4 are true, because speciation of polyploidy happen when diploid cell undergo failed meiosis, then producing diploid gametes, then when self-fertilized to produce tetraploid zygotes. But producing triploid gametes will again in the next generation may cause sterile progeny also. So this is not the adequate answer here. So option is only 1, 2 and 4 are true. Next question, F1 seeds are used for the cultivation of options hybrid varieties, composite varieties, both A and B, and none of the above. Answer is both A and B, because for production of hybrid variety as well as composite variety we generally use F1 seeds. In composites we generally compose the different F1 seeds. Next is in the condition of epistasis. Options are single gene influences one trait, more than single gene influences one trait, single gene influences more than one trait, or multiple genes influence multiple traits. Here the answer is single gene influence more than one trait. It is also the condition of pleiotropy. Next question is, which is true about triticale? Options are it has high degree of fertility. That is false. If it was so, triticale would have been popular by this time and cultivated in majority of the area worldwide. Next option, it is amphidiploid with 54 chromosomes. Not so all the time, and it is not amphidiploid because there will be alloploidy situation where we'll use the crossing of wheat into rye. Wheat is already an allopolyploid, and rye again it is crossed with wheat, so diploidy is not possible here. Third option is it is a cross between sickle cereal and triticum vulgar. Then none of the above. Answer here is it is a cross between wheat and rye. C is the option. Okay. Dash is responsible for poor fruit set in custard apple. Custard apple is nothing but your sitafal. Options are self incompatibility, heterostyly, both A and B, dichogamy. Answer here is dichogamy. Dichogamy is a situation where ripening or maturity of stamens and pistils at different times. So this happens in custard apple. Next question is, NADPH H plus is formed in cyclic photophosphorylation, non-cyclic photophosphorylation, pseudo-cyclic photophosphorylation, none of the above. Though this is a physiological based question but frequently asked in our plant science exams also. So the answer is non-cyclic photophosphorylation. I am not going to explain you that particular cycle and all. You just read it. Next question is the method of selection of plants from population by their phenotype. Read it carefully. Method of selection of plants from population by their phenotype. Phenotype, when it describes the population, the answer is mass selection, because in case of pure line selection we majorly go for phenotype, but homozygosity and homogeneity attention will be the major concentration, and especially this is used in self pollinated crops, whereas in mass selection we generally uh, concentrate on homozygosity but not homogeneity. Okay. Then clonal selection means it's a kind of asexual reproduction where we'll go for cloning or production of identical copies, either it may be gene or any tissue. Next is bacteria protect themselves from virus by fragmenting viral DNA with options are ligase, endonuclease, exonuclease, and gyrase. Here, when you think about these options, you should know the particular functions. When it comes to gyrase helps in negative supercoiling of the DNA. Ligase is for joining the breaks of phosphodiester bond in DNA, which helps in DNA repair, damage mechanisms and all. Then the function of exonuclease is proofreading during the DNA polymerization process? So the answer here is endonuclease, because bacteria protect themselves from virus by fragmenting viral DNA with endonuclease. What endonuclease do here? It will give the internal cuts so that viral fragments won't get into the bacterial circular DNA fragment. Okay, that is the thing here. Next question is, the vaccines prepared through recombinant DNA technology are options: third generation vaccine, first generation vaccine, second generation vaccine, none. Recombinant DNA technology, remember, first and second generation vaccines were prepared by using some animals, some marine organisms and all. But the third generation vaccines are purely dependent on recombinant DNA technology. Which of the following is the quality of improved transgenic basmati rice? Options are: gives high yield but no characteristic aroma, gives high yield and is rich in vitamin A. Third option: does not require chemical fertilizers and growth hormones, then resistant to insects and diseases. Quality of improved transgenic basmati rice. This again shows finger towards golden rice. So golden rice is for high yield and rich in vitamin A. Many other approaches tried in transgenic basmati rice, but golden rice is one of the success stories. Though now the golden rice production is not that much popular, but many of the countries worldwide adopted this for cultivation, not on a large scale but up to some extent. So appropriate answer is high yield with rich vitamin A. Next question, first clinical application of gene therapy over a 4-year-old girl was for. Options are adenosine deaminase deficiency, adenosine deficiency, growth deficiency, or adenine deficiency. Answer here is adenosine deaminase deficiency. Here you should know what is this particular condition. Adenosine deaminase, which damages the immune system, disables all the immune protection. That means if you are with this particular deficiency, means you will be susceptible to all fungal, bacterial and viral infections. Okay, so gene therapy was done on 4-year-old girl in order to overcome this. Next question, excision and insertion of a gene is called as excision, means cutting, insertion means inserting. Options are biotechnology, genetic engineering, cytogenetics and gene therapy. The answer here is genetic engineering, because in gene therapy different approaches are used, but excision and insertion, two options are mainly used under genetic engineering. And again genetic engineering comes under biotechnology only, but here specifically when they have asked, we should give specific answer. Answer is genetic engineering. Next question, sum total of breeding values of individuals in a random mating population is 0, unity, infinite and can't calculate. The answer here is 0. So you should know what is breeding value. Breeding value is the sum of breeding potentiality of allele containing in the genotype, means how potential it is to contribute to the next generation. So when it comes to random mating population, this is 0, because in random mating population we cannot product the zygosity. Okay. Next question, which of the following ordinate is valid in a normally distributed variate or curve where 95% of the total observations vary in the range of population mean and only 5% deviate statistically from population mean? So to understand this, the options here are mu, then sigma, mu plus sigma, then mu plus 2 sigma. Okay, these are the options. Now to understand this, first we should draw the normal distribution count, this one curve. Here there are three situations. One is leptokurtic, mesokurtic and platykurtic, as you know. Okay, three will be there, steep curve will be there. So this is leptokurtic. This is mesokurtic, and this last flat one is platykurtic. So what we expect, 95% of the total observation vary in range of population mean, and only 5% deviation statistically. Only 5% deviation comes under leptokurtic only. So the answer here comes mu plus 2 sigma. Okay, that is for leptokurtic situation. Next, which of the following is not true for inbreeding? It causes inbreeding depression after few generation, leads to homozygosity. It is used to produce a pure line. It is always increase the productivity. Not true, they have asked. Inbreeding does not increase any productivity. So fourth one is the option D is the option. Okay. 5-GAA-TTC3- is a restriction site of a widely used restriction enzyme which produces sticky ends. The enzyme is, options are BAMH1, HIND3, ECOR1 or HIND2. Answer here is ECOR1 because they have asked the restriction enzyme for producing the sticky ends. As you have known, when where we use the restriction enzymes, that is to make either sticky ends or blunt ends. BAMH1, HIND3 as well as HIND2 are for making the blunt ends, whereas for sticky end making, ECORI1 will be helpful. Next question, spherical to ovoid fruit of potato is called. Options: berry, ball, capsule, all the above. Here you can call it with any name. Either it may be berry, ball or capsule, whatever it may be. All the above is the option. Early varieties of field peas are Pantmatar, Urkel, early Badger, all the above. So early varieties of peas are all the above is the option. The constraints encountered in mango hybridization are, options are heterozygous nature, long juvenile phase, polyembryony, all the above. Answer is all the above. Onla variety developed by selection are NA4, NA7, Anand 2, all the above. Answer is all the above. The primitive varieties which evolved without a systematic and sustained plant breeding effort. Okay. Here what they have asked, primitive varieties means very old varieties evolved without systematic and sustained plant breeding effort. If we are using this systematic and sustained plant breeding efforts means, again we may get absolute varieties or breeding lines or all those things, but here the answer is land races because primitive varieties are always land races. Okay. Multicut varieties of fodder sorghum. Options: Ruchira or Maldandi, Harsona 855, Pantchari 5, all the above. Answer is all the above. Next, dual purpose variety of fodder sorghum. CSH 13 R hybrid, CSV 15, then SPV 669, all the above. Answer is all the above. Fodder maize variety developed. This is not required. Berseem crop is generally a self-pollinated, cross-pollinated, often cross-pollinated. None of this. Berseem is cross-pollinated. Salinity tolerance variety of sugarcane is CO 453, CO 62125, both A and B. None of the above. Both are salinity tolerant varieties. Performance of genotype with respect to changing environmental factors over time within a given locality is referred to. Options are stability, adaptability, adaptation, all the above. Answer here is stability. You should know the definition to answer these kind of questions. Here they have asked performance of a genotype with respect to changing environmental factors over time within a given location. In a location, if a variety is performing better for a longer period of time means that is giving the stable performance. So stability is the option. When it comes to adaptation, adaptation का definition क्या होता है? It is fitting to the given environment. Means how the if immediately you are going to introduce one particular variety, whether it is performing better in a given situation or not, that is nothing but adaptation. Adaptability means it takes time, over a given period of time, how it is showing the performance. Okay, so these three definitions are very important. Again, in adaptation there are three things: structural, behavioral, physiological adaptation and all. Next question is genetic buffering capacity of a genotype to environmental fluctuations is. Answer, the options are genetic homeostasis, physiological homeostasis, both or none of the above. Genetic buffering capacity, when it defines genetic homeostasis, okay, homeostasis, that is option A is the answer. Next question, safflower hybrid based on genetic male sterility are Nari H15, DSH9, MKH2, all the above. Answer here is all the above. First castor hybrid GCH3 in India is a cross between VP1 4 into 48-1, TSP10R into JI15, VP1 into TSP10R, or none of the above. Answer here is TSP10R into JI15. Okay, next question. Rabi sorghum hybrid seed production plots should have minimum field inspections. Remember, sorghum is often cross-pollinated crop. In under often cross-pollinated and cross-pollinated crops, the field inspections are more than 3 or 4. Okay, if paddy means 2 or 3 are enough, means in self-pollinated crops less number of field inspections are sufficient, but when it comes to often cross-pollinated or cross-pollinated, we need more inspections. So here the answer is 4. So friends, thank you. If you have any doubt or if you want video or information on specific topic, please keep it in the comment section. Please keep on liking, subscribing and commenting. Thank you once again.